Imagine you're looking out of an airplane window at night, right? Down at this massive, sprawling metropolis.
Mm.
And if you were to roll down the window, assuming you could somehow survive doing that at 30,000 feet, you would just hear this collective roar of the city.
Yeah, just absolute noise.
Exactly. You'd hear the combined hum of, like, traffic, sirens, industrial factories, just millions of overlapping lives all at once. And for decades, when scientists wanted to understand the microbiome, the trillions of microbes living in and on you, that collective roar is exactly what they had to listen to.
That's a really great way to put it, actually.
Right, because they would take a biological sample, essentially drop it into a blender, and just sequence all the DNA at once. It's called metagenomics, or bulk sequencing. You get the overall noise of the metropolis, but you have zero idea who is living in which apartment, or who is saying what to whom.
You lose all the context.
Exactly. But what if we could change that? What if you could, I don't know, glide down from the clouds, hover outside a single window on the 40th floor of a specific building, and just listen to the real-time private conversation of one resident without ever knocking on the door or removing them from their living room?
That visual perfectly captures the scale of the technological leap you're exploring today. We are really transitioning from deciphering that blended roar of a microbial city to basically eavesdropping on the whispers of a single localized individual.
Which brings us to the mission of this deep dive. Today, we are unpacking a genuinely groundbreaking 2026 research paper published in Cell Genomics by Lon and colleagues. It's titled Microbial Single-Cell Omics in Situ.
A huge paper.
Yeah, massive. We really want to understand how researchers are finally reading the exact genetic blueprints and, more importantly, the real-time active thoughts of individual living bacteria right where they sit in our tissues.
The paradigm shift here really cannot be overstated. I mean, historically, interrogating bacterial populations left you with two highly flawed options, right? You either blend the tissue up, which immediately destroys all that spatial context.
Right, you completely lose the architecture of the apartment building.
Exactly.
Yeah.
Or you attempt to use droplet microfluidics to isolate single cells.
And droplet microfluidics just doesn't cut it for this. I mean, I know relying on a Poisson distribution to randomly trap single cells into tiny fluid droplets is, well, it's inherently inefficient. You end up with mostly empty droplets.
Mostly empty.
But bacteria present a completely different set of biological hurdles compared to, say, human cells. They contain orders of magnitude less DNA and RNA. Plus, eukaryotic sequencing technologies generally rely on grabbing those long collier tails attached to human messenger RNA to amplify the signal, and bacterial mRNA usually just lacks those convenient handles.
Precisely. You are trying to fish for a microscopically small, rapidly degrading signal, and you just don't have the proper hook. Because of this, scientists desperately needed a completely novel physical mechanism to isolate and sequence individual bacteria directly from complex environments without obliterating the neighborhood in the process.
Okay, let's unpack this. So the researchers developed a system called LIFT, which stands for Laser-Induced Forward Transfer. It essentially functions as a highly precise, laser-powered claw machine.
Yeah, LIFT is this remarkable intersection of optics, thermodynamics, and biology. The process starts by placing a biological sample, so perhaps a smear of human saliva or microslice of gut tissue onto a specialized quartz slide.
Okay.
The slide is completely optically transparent, but it is coated with an ultra-thin film of aluminum, exactly 25 nanometers thick.
Wait, 25 nanometers. So the aluminum is acting as this, like, localized sacrificial shock absorber.
That is a highly accurate way to frame it, yeah. You observe the sample through a microscope, target a single bacterium, and you fire a 532 nanometer nanosecond pulse laser from below the slide.
Come look on the bottom.
Right. Because the quartz is transparent, the laser passes right through it. But the energy is immediately absorbed by that 25 nanometer aluminum layer right beneath the target cell. In a fraction of a nanosecond, the aluminum undergoes a rapid phase change. It vaporizes and expands into a microscopic, localized thermal bubble.
Which creates kinetic energy, acting like a microscopic catapult to just propel the cell upward.
Yes. The bubble gently ejects the selected microbe into a tiny receiver tube, hovering just microns above the sample. And the engineering tolerance here is incredibly tight.
I can imagine.
Yeah, the researchers drive this mechanism with specific energy levels, tightly bounded between 60 to 75 nanojoules.
Okay, so what happens if you miss that window?
Well, if the pulse is too weak, say 40 nanojoules, the kinetic bubble just isn't large enough. The cell basically vibrates and remains stuck to the surface. But if the energy is too high, the sheer mechanical stress completely ruptures the bacterial membrane, spilling all its genetic contents before it even reaches the tube.
Oh, wow. So it has to be exactly 60 to 75 nanojoules.
Exactly. At that energy, the spot size is perfectly calibrated to pop the cell out intact, leaving all the neighboring cells entirely undisturbed.
But wait, I have to push back on the practicality of this for a second. If you were examining a highly complex, messy sample like dental plaque or gut mucus, laser catapulting individual cells one by one sounds, well, painfully slow and tedious. If the goal is mapping a microbiome, isn't this throughput terrible compared to just bulk sequencing a swab?
It is slower, yes.
And furthermore, how do you even know you're aiming the laser at a bacterium and not like a tiny fragment of a dead skin cell or a microscopic piece of food debris?
Right, so the throughput is undeniably lower than bulk sequencing, but it trades sheer volume for unparalleled spatial resolution and absolute purity. You aren't guessing who is present. You are definitively linking a physical location to a specific genome.
Okay, that makes sense.
And to address your second point about misidentifying debris, because that was actually the primary technical hurdle, the researchers solved it by integrating a secondary non-destructive optical system before the lift laser is ever fired. It's called Raman spectroscopy.
Ah, I love the physics of Raman spectroscopy. It sounds like something straight out of a sci-fi novel where you just scan an object to determine its chemical makeup.
It really does. It relies on a phenomenon called inelastic scattering. Before they arm the catapult laser, they shine a continuous 3-milliwatt laser onto the target for exactly 3 seconds. When photons from this laser hit the target, the vast majority of light bounces back elastically, meaning it has the exact same energy and color. That's called Rayleigh scattering. But about 1 in 10 million photons will interact with the actual vibrational energy of the molecular bonds inside the target.
So depending on whether the photon hits like a lipid, a protein, or nucleic acid, the chemical bonds absorb a tiny fraction of that energy, kind of like springs compressing and releasing.
Exactly like that. Because some energy was absorbed by the chemical bond, the photon that bounces back has a slightly lower energy and therefore a slightly shifted wavelength or color.
Wow.
Yeah. By measuring those microscopic shifts in the returning light, the system generates a Raman spectral fingerprint. It's this highly complex, completely unique graph of molecular vibrations. It definitively confirms this object contains the complex biochemical signature of a living microbe, and not, say, the simple lipid profile of a leftover potato chip.
So you scan the object, verify its chemical ID badge with Raman spectroscopy, and only then do you fire the 75 nanojoule laser to blast it into the collection tube.
That's the sequence, yes.
The elegance of that dual laser setup is really validated by their initial saliva experiment, isn't it? I mean, they visually located a single megamonas funiformis cell in a human saliva sample. Raman scanned it, lifted it, and when they sequenced the single cell's DNA, they achieved a draft genome with 99.99% completeness and a staggering 0.33% contamination rate.
Achieving that level of genomic purity from a single cell, especially one extracted from a dense, complex microbial community, just proves that the lift mechanism is practically flawless. It cleanly physically isolates the target without dragging all the surrounding biochemical noise along with it.
So just to do a quick recap for everyone listening, we've covered the limitations of old microfluidics, the mechanics of this lift-T laser ejection system, and how they use Raman spectra as a sort of chemical ID badge. And it makes you think, if you had a laser tweezer that could sequence any single microscopic organism around you without removing it from its home, where is the first place you'd point it?
It's a great question.
Right. But so we have essentially built the perfect microscopic trap to isolate a single microbe without destroying its home. But capturing the blueprint is only half the battle. The true leap in this paper comes when we shift from looking at static DNA to observing how these bacteria actively behave when their environment turns into a literal war zone.
Yes, this is the transition from genomics to transcriptomics, reading the single-cell RNA or scRNA. It allows us to read the active genetic instructions the cell is deploying in real time.
The search history, basically.
Exactly. To test this, the researchers isolated a strain of Bacillus licheniformis from human saliva, cultured it, and subjected it to severe environmental stress by dropping the pH with citric acid for 9 days.
They were trying to force the population into sporulation, right, where a bacterium facing starvation, or in this case extreme acid, initiates a massive biological overhaul to turn itself into a dormant, heavily armored spore just to survive the famine.
Yes, but the cRNA sequencing revealed that this genetically identical population didn't just behave as a monolith, By day 3 of the acid stress, the population had fractured into 5 distinct morphological types, and each was running a completely different survival algorithm.
Right. Looking at the transcriptomic data for type I cells, there is a massive spike in the expression of the MANA gene and various FDHFY-DP family oxidoreductases. So these elongated, rod-shaped cells are the fighters. They are aggressively ramping up sugar transport and metabolic pathways, trying to basically physically out-metabolize the acid and keep the cellular factory running.
Building on that, the type 2 cells displayed a slight physical indentation in the middle of their structure. Their cRNA showed an overwhelming expression of helicase genes, the chromosomal protein DNAi, and DNA polymerase.
Which means they are stubbornly rushing to finish replicating their DNA and divide before the environment becomes completely unsurvivable. They are like the factory workers ignoring the fire alarm to just finish the final product.
That's a good analogy. We can skip over type 3, which were essentially transitioning short rods, and look closely at type 4.
Yeah, by type 4, I see a sharp upregulation in elongation factors and GTP-associated genes. Since we know they are physically forming the spore inside the mother cell at this stage, they are actively translating the final structural proteins to build the physical armor. They are packing their bags and building the bunker.
And finally, the type V cells are the mature, fully spherical spores. The factory is officially shut down. Yet, paradoxically, their cRNA showed they were highly expressing specific transporter genes, like NAC, alongside bacteriocins.
Which seems super counterintuitive. If you are a dormant, armored spore, why are you actively running transport protein?
It actually acts as an active defense mechanism to block premature germination. The spore is constantly monitoring the exterior environment, ensuring it doesn't accidentally drop its armor and wake up while that lethal citric acid is still present.
The division of labor is just incredible, but here's where it gets really interesting. While tracking this cellular panic, the LIFT cRNA data captured a highly specific anomaly regarding bacterial poly-A tails, the poly-A paradox.
This is a fascinating part of the paper.
Because, in human biology, when our cells transcribe messenger RNA, an enzyme adds a long tail of adenine nucleotides, a poly-A tail, to the end of the strand. It acts as a protective cap, preventing the RNA from degrading before the ribosome can actually read it. So finding these protective tails on bacterial RNA during a stress event seems entirely contradictory.
What's fascinating here is how evolutionary biology rarely relies on a single universal rule. In eukaryotes, you're correct. A poly-A tail, which is often hundreds of nucleotides long, is a stabilizing shield. But in bacteria, evolution totally inverted the mechanism. A stretch of adenylates on a bacterial mRNA transcript is actually a chemical kiss of death.
Wow.
Yeah, it acts as a highly specific homing beacon that actively summons exonucleases, basically cellular Pac-Men, to latch onto the RNA and aggressively destroy it.
So they are deliberately tagging their own genetic instructions for destruction. I guess because in a rapidly shifting hostile environment, holding on to outdated survival instructions is just a lethal waste of resources.
Exactly. Bacteria need to pivot their metabolic strategies instantly. They add a short poly-A tail to immediately terminate and recycle the old RNA molecule so they can clear the cellular machinery to print new stress-appropriate instructions. And because the LIFT technology captures the RNA in situ at high fidelity, the researchers actually caught this degradation happening in real time.
Yeah, they isolated the transcripts for the TXTA gene, which codes for a citrate transporter, the specific protein handling the citric acid the bacteria were being flooded with.
Right.
And the cRNA data captured partial, heavily degraded transcripts of this gene with the poly-A tails actively attached. They could literally map the exonucleases shredding the document nucleotide by nucleotide from the 3' to the 5' end. We are watching a single bacterium shred its own internal documents in real time just to adapt.
It is a profound validation of the technology's single nucleotide resolution. Older transcriptomic techniques often artificially force long poly-A tails onto bacterial RNA just to use eukaryotic amplification kits. Doing so completely masks this natural, targeted biological degradation process. We would have never seen the shredder in action.
That's wild. Observing a genetically identical population deploy 5 completely different survival strategies under the exact same stress is just a fascinating mirror to complex systems. Just to quickly recap this part for you listening, we tracked the step-by-step algorithm of sporulation in Bacillus licheniformis, and we uncovered the paradoxical role of bacterial poly-A tails marking RNA for destruction. And Think about how different individuals in your own workplace or family deploy completely different algorithms during a crisis. Some fight the problem, some stubbornly keep working, and others immediately build a bunker.
It's a very human parallel.
Right. But watching cultures panic in an artificial petri dish is really just the warm-up. To realize the true clinical potential of LIFT, we have to move into the ultimate frontier, reading the diaries of bacteria embedded inside highly complex, living host tissues.
And moving from in vitro to in situ introduces massive background noise. The researchers first utilized a mouse gut model. The fundamental problem here is locating a microscopic, transcriptionally active bacterium buried within millions of host epithelial cells and dense layers of gut mucus.
So you have to find a way to make only the active bacteria glow without lighting up the entire mouse tissue. And they solved this by feeding the mice highly specific fluorescent D-amino acids, specifically molecules tagged as Cy3A and FAMIDA.
The biochemistry behind this is brilliant. So amino acids are the building blocks of proteins, and they exist in two structural orientations, or chiralities: left-handed L-amino acids and right-handed D-amino acids.
And human and mouse cells almost exclusively utilize left-handed amino acids for their cellular architecture. They completely lack the transpeptidase enzymes required to stitch right-handed D-amino acids into their structures. So to a human cell, a D-amino acid is just useless, invisible debris.
But bacteria uniquely rely on right-handed D-amino acids to construct and repair their thick peptidoglycan cell walls. By feeding the mice these glowing D-amino acid bricks, the host tissue ignores them entirely. But any bacterium actively maintaining its cell wall immediately absorbs the fluorescent molecules and begins to glow.
So they created a highly specific molecular beacon. They took a micro slice of the mouse gut, zeroed in on the glowing fluorescent spots, confirmed them with the Raman laser, and lifted them straight out of the mucus.
Which is an incredible feat on its own.
Yeah, and they successfully sequenced typical gut flora, like Prevotella copri, proving they could pull high-quality sRNA from a living tissue matrix. But the absolute mic drop of this paper comes when they applied this to human clinical pathology. They secured a tumor biopsy from a 50-year-old patient with a long history of smoking who presented with lung metastasis and colorectal cancer.
Using standard antibody staining to map the tissue, they located microbes distributed directly on the mucosal surface of the colorectal tumor itself.
Okay.
They identified a specific rod-shaped bacterium, verified its microbial signature with Raman spectroscopy to ensure they weren't, targeting a human cancer cell, and then lifted it. Because they were flying blind here, they had to perform a de novo transcriptome assembly, meaning reconstructing the active RNA sequences from scratch without a predetermined reference genome.
And what did they find living on the surface of this patient's colon tumor?
The genome assembled into Bacteroides xylanosulfans,
Which is a known gut microbe. But the critical question is, what was its search history? By reading its real-time RNA, what did the LIFT technology reveal this bacterium was actively doing while anchored to a malignant tumor?
The cRNA profile was astonishing. The bacterium was actively overexpressing a specific gene known as NICS. The NICS gene encodes an enzyme responsible for nicotine degradation.
In a patient with a 50-year history of heavy smoking.
Exactly. And it gets deeper. The transcriptomic data also showed massive upregulation of specific membrane proteins, notably the Syscrag-A family, various ABC transporters, and porins. These are highly complex molecular machines designed for the massive uptake of large nutrients. In the specific context of the colon's mucosal lining, these proteins are the primary machinery for mucin metabolism.
Okay, so if we synthesize all of this LIFT allowed the researchers to bypass the vague guesswork of bulk sequencing. They didn't just note that Bacteroides was somewhere in the patient's gut. They basically kicked down the door, caught the bacterium physically anchored to a human tumor, and read its genetic diary to discover it was actively metabolizing the host's protective mucus layer for energy, while simultaneously deploying enzymes to clean up the highly toxic nicotine left over from a lifetime of smoking.
That's exactly what they saw. If we connect this to the bigger picture, the implications for precision medicine are staggering. We're no longer limited to analyzing the static presence or absence of a microbe. We can directly observe the real-time metabolic negotiations happening at the disease site. Yeah. We can measure the precise physiological toll the microbe is taking on the host, like degrading the mucin barrier, or identify the environmental remediation it is attempting, like clearing out accumulated carcinogens.
I mean, imagine the personalized therapies of the future. Instead of bombarding your system with broad-spectrum antibiotics, a doctor could take a biopsy, read the real-time search history of the microbes on your tissues, and deploy targeted treatments based on exactly what metabolic pathways those specific microbes are running at that exact second.
It's incredible.
So just to give you a quick recap of this final leg, we moved into actual tissues, using D-amino acids to light up active bacteria in mice, and then isolated a specific bacteroid cell in a human colorectal tumor to catch it actively processing nicotine. And it makes you wonder if doctors could read the real-time search history of the microbes sitting on your own tissues. What kind of personalized treatments could they design for you?
The potential is really limitless.
So what does this all mean? We started this deep dive staring down at a massive, noisy metropolis from an airplane window, forced to settle for the blended, chaotic audio of metagenomics. We ended armed with an incredibly elegant 532-nanometer laser claw machine that allows us to zoom down to a single biological apartment firmly attached to a human cancer cell and read the exact real-time genetic thoughts of a single microscopic organism.
This raises an important question about our fundamental understanding of the human microbiome. Metagenomics was essentially a census. It told us who was legally registered to live in the city. But this lift single-cell omics technology tells us exactly what those residents are whispering to each other, how they radically alter their survival algorithms under stress, and precisely how they are manipulating our human cells at the molecular level. It elevates our understanding from a static photograph to a dynamic, high-definition movie of microbial life.
And that leaves us with a profound final thought for you to mull over. We just observed a single bacteroid cell actively attempting to process and degrade the toxic legacy of nicotine in a smoker's colon. As technologies like LIFFT continue to scale, we have to fundamentally rethink our relationship with our biome. Are the microbes embedded in our tissues just passive hitchhikers along for the ride, or are they acting as a dynamic, highly responsive, real-time pharmacy constantly deploying novel genetic algorithms to manage and perhaps even cure the toxic microenvironments we unwittingly create for them? Keep diving deep, and we'll catch you next time.